Well, good morning, everyone. First of all, I'd like to apologise for the fact that a relatively late change in my plans meant that this needs to be pre-recorded. I'm very sad about that, particularly missing the opportunity to discuss today's talk with you at the end of the presentation. But what I wanted to talk to you today about is how my laboratory has been studying a particular human genetic disorder that causes structural brain defects at birth. And it's a condition known as holoprosencephaly, which will often be abbreviated to HPE on my slides. So I'm sure I don't need to remind this audience that the mammalian forebrain is always divided into left and right hemispheres. And this process of doing so occurs early in embryonic development. And you can see here that it is complete in the human fetal brain by halfway through gestation. Now, the condition I'm talking about today, or holoprosencephaly, occurs when this process of forebrain division either doesn't happen or happens only partially. So this picture of a brain was taken at postmortem from a fetus with holoprosencephaly, which died in utero. And you can see that the forebrain has no separation whatsoever. Now, this is a severe case of HPE, but one of the striking clinical features of human holoprosencephaly is that it has extreme phenotypic variability. So in addition, before we look at the next slide, you need to know that the same event that causes the brain to divide into left and right hemispheres actually controls the formation of our face as well. And in particular, it affects the middle of our face. So this means that craniofacial defects generally, but not always, accompany the holoprosencephaly brain defects. So these are images of people who have holoprosencephaly, and you can see that some people with HPE have only very mild craniofacial defects. For example, this woman just has a single central incisor. This child has eyes that are unusually close together, and this child has a cleft palate, whereas this child, who is stillborn, has a single eye with a proboscis, and this proboscis is the remnants of the forebrain. In fact, this variability is so large that clinicians tend to divide holoprosencephaly into several categories that depend upon the extent of the structural defects of the brain, but which often correlate with the severity of the craniofacial defects. But what is interesting is that there's no simple relationship between the mutation carried by an individual and the phenotype of that individual. And this is demonstrated in this family, which was included in the first report of a gene associated with holoprosencephaly. This gene is called sonic hedgehog in both man and mouse, and this work was published in 1996. So in some families which had heritable mutations in the sonic hedgehog gene, this inheritance was not strictly Mendelian. For example, here this family illustrates variable expressivity, in which the same mutation leads to different phenotypic consequences in different individuals. This is the woman you saw on the earlier slide, and she has a very mild presentation of holoprosencephaly, whereas 2 of her 3 children had more severe presentations, including this child here with cleft lip and palate. But the DNA analysis shows that each of them has the same sonic hedgehog mutation. And another example of this is an extreme form of variable expressivity is when an individual carries a mutation but doesn't present with a disease at all. And this is called incomplete penetrance. And you can see in this 4-generation pedigree that some of these siblings, shown here within the box, this includes 2 brothers, both of which carry the HPE-causing variant in the sonic hedgehog gene, but only one of which presented with the disease. So both of these are non-Mendelian features of inheritance. And this diagram illustrates our current thinking about the etiology of inherited disease. So we generally think that some diseases have a very strong genetic component, and we call these Mendelian diseases. In these diseases, there is a strong correlation between a gene variant and phenotype, and these diseases are generally rare. Other diseases are multifactorial. Either they involve multiple genetic factors or a combination of genetic and environmental factors that interact. And this causes a variable relationship between gene variants and the disease phenotype. So these diseases tend to occur more frequently within the population. So based on what I've told you about HPE heritability, you'd think that HPE occupies a space somewhere in this region on this diagram. But HPE is very rare and has a frequency of 1 in 10,000 at birth. Now, this apparent discrepancy can be resolved by some human genetics observations from the 1970s, in which the in utero frequency of HPE was estimated to be 1 in 250, but the majority of HPE cases die in utero. So in this case then, HPE is actually relatively common, and this fits precisely with the heritability aspects that we have discussed. So in terms of the genetic etiology then, we'd expect HPE to be multifactorial and perhaps have both genetic and environmental aspects. So in the last 2 decades, human geneticists have used increasingly sophisticated genome analysis techniques, and we now know a lot more about HPE genetics than we did when sonic hedgehog was first identified as a causative gene in 1996. So thus, in humans, our current understanding is that there are 4 genes, at least one of which is mutated in more than 95% of the solved HPE cases with normal chromosomes. And each of these 4 is considered a common or major effect HPE gene. In addition, a growing number of genes have each been found to be mutated in a very few cases of HPE. And putting all of this information together, it seems that some cases of HPE arise by a combination of multiple rare small effect alleles, perhaps in combination with an environmental insult. Even so, even with all of this genetic knowledge, our genomic sequencing of holoprosencephaly patients still only solves 20% of HPE cases with normal chromosomes. So many of the genes associated with human HPE act within the sonic hedgehog signaling pathway diagrammed here. So, for example, CIX3 and TGIF are involved in the control of sonic hedgehog gene expression, and many of the uncommon mutations are found in molecules involved in sonic hedgehog production, release from the tissue that sends the sonic hedgehog signal to the brain and face, and the reception of the sonic hedgehog signal by the cells of the face and brain. But many of the other genes that we saw on the previous diagram don't act in the hedgehog pathway, and this suggests there's much to learn about HPE mechanisms. So to study this, my laboratory has used the mouse as a model system. And we're able to do this because mice also get holoprosencephaly. And indeed, so far, every gene that's known to cause HPE in man also does so in the mouse when that has been tested. So this indicates that the process of patterning of the face and brain is highly conserved between mouse and man. And we think that the mouse is a very good tool for studying HPE etiology and pathogenesis. So this slide shows different things that we've used mice to understand about HPE. And in the remainder of the talk, I'll just show you some examples from our work in these areas. So one thing that the mouse models are useful for is to understand the biological process by which a particular disease arises. Now, you may recall that HPE occurs when structures at the middle of the embryo fail to develop properly. And this is because during normal development, a group of cells that lie at the center of the embryo send signals to the overlying tissues that pattern, and these signals pattern these overlying cells. So together, the midline cells that I've shown here in reds and pinks are called the midline mesoderm. And the midline mesoderm is given different names according to where it lies on the AP axis of the embryo. So the midline mesoderm that underlies the forebrain is called the pre-chordal plate, and that just behind it is the anterior notochord, and this underlies the rest of the brain. And then the notochord tissues, the midline mesoderm tissues that underlie the trunk, is called the notochord. This shows another diagram of the developing embryo alongside a mouse embryo that has been dissected at 8.5 days post-coitum, and the embryo has then been flattened out, and we're viewing it dorsally. So this mouse embryo is showing the expression of the sonic hedgehog gene, and you can see that this gene is expressed in all of the axial mesoderm. And this is the secreted molecule that sends a signal to pattern the overlying tissues. So in essence, holoprosencephaly occurs when the sonic hedgehog signal is not sent from the axial, from the midline mesoderm, or when the sonic hedgehog signal is not received by the overlying developing face and brain. And I just want to come back and remind you that in terms of HPE, it's sonic hedgehog signal being sent from the pre-chordal plate that is essential to pattern the ventral part of the developing forebrain. So here's another view of the pre-chordal plate, this time in some beautiful SEM images of the mouse, in which the mouse has been cut in half, and then the relevant tissues false coloured here. So this here is an early somite stage embryo, and here you can see the pre-chordal plate and the way in which it abuts the neurectoderm, and in particular the future forebrain neurectoderm. And one of the things that we know from analysing mouse mutants is that the sonic hedgehog signal within this pre-chordal plate tissue is sent to the overlying neurectoderm in order to cause correct ventral forebrain patterning by the 3 somite stage. So some years ago, my laboratory conducted a phenotype-driven genetic screen for genes that cause holoprosencephaly, and we discovered this particular mouse strain. And we called this mouse strain Kumba. It gets abbreviated to KU. So embryos homozygous for this mutation display a severe form of holoprosencephaly, and this is indicated by the cyclopic eye, which is buried here within the centre of the head, and thus is best seen in this section. And in addition, you can see that these embryos have a proboscis here, that, as in the human cases, this is the remnants of the forebrain. So molecular analysis showed that these mice carry a severe loss of function mutation in the ZIC2 gene. So as I've already said, midline development is highly conserved, and ZIC2 mutation also causes holoprosencephaly in man. Indeed, ZIC2 is one of the genes most commonly mutated in human HPE cases. Now, the ZIC2 gene encodes a transcription factor. The defining feature, which I've shown you a line diagram of across the top here, the defining feature of this transcription factor is a zinc finger domain that consists of 5 tandem C2H2 type zinc fingers. And this domain is responsible for DNA binding as well as protein-protein interactions. Now, the rest of this slide shows you the mutations that have been associated with human HPE. And you can see that the mutations span the whole protein and are of all types. But the missense mutations, shown here in triangles, tend to be clustered within the zinc finger domain. So the Kumba mouse mutant has a missense mutation in a canonical cysteine within the 4th zinc finger. And indeed, there has been a human patient now that's been identified that has precisely this mutation. So we decided to use this mouse strain to understand exactly how ZIC2 mutation causes holoprosencephaly. So you may recall that most mutations that cause HPE affect the sonic hedgehog signaling pathway. So at the time that we discovered this mouse strain, human clinicians assumed that ZIC2 must act as a transcription factor in the sonic hedgehog pathway, so acting in the receiving cell to control the expression of sonic hedgehog target genes. And there's biochemical data that suggests this is a likely scenario, since ZIC2 can bind to Gli molecules, which are the well-known transcriptional mediators of sonic hedgehog signaling. And they bind to one another because their DNA binding domains are very similar. So we, however, thought this was somewhat unlikely because ZIC2 is not expressed in the receiving tissue. So ZIC2 is not expressed in the ventral forebrain, and we set out to investigate pre-chordal plate development in the ZIC2 null embryos. So in each of the next series of pictures, you can see a wild-type embryo on the left and a stage-matched mutant embryo on the right. In each case, the mRNA expression of particular genes shown by whole mount in situ hybridization. So in the late streak stage mouse embryos shown here, the pre-chordal plate cells have already differentiated and migrated to the anterior of the embryo. And here they're marked by the expression of a gene called H-hex. So this picture shows that in the ZIC2 mutants, the pre-chordal plate cells have differentiated and migrated as expected. But strikingly, expression of this gene is specifically downregulated in a region of the embryo called the node. Now, the cells that pass through the node at this time should differentiate into anterior notochord. And so what we did next was to look for transcripts of other genes expressed in the node. And we find that each of these genes is depleted in the node as ZIC2 mutants. So HX, FOXA2, Cerberus, and all of these other genes involved in cilia formation in the node are all downregulated. And it seems as though ZIC2 is a master regulator of gene expression in this tissue just at the stage that the anterior notochord differentiation is occurring. So I'm just going to go back and draw your attention here to FOXA2 expression, since we know that embryos that are null for FOXA2 don't produce the midline tissues, or the midline mesoderm. So if you look at if you follow FOXA2 expression in ZIC2 mutant embryos over time, you can see that at early stages, its expression is unaltered in the mutant embryos. And this is the stage at which the pre-chordal plate cells are being formed. But as we've seen, once the node begins to form, FOXA2 expression becomes downregulated. And it's these cells that should give rise to the anterior notochord. And as you can see in these early bud stage embryos, the notochord cells that are marked here by FOXA2 expression are missing. But the real surprise came when we looked at slightly later embryos, because now FOXA2 is expressed also in the pre-chordal plate. And you can see in the mutant embryos that not just the cranial notochord cells, but also the pre-chordal plate cells are missing. So this led to our conclusion that ZIC2 function is required in the late streak node to direct cranial notochord differentiation. And it seems that the cranial notochord is necessary for survival of the pre-chordal plate. And this was supported by two other pieces of data. So first of all, ZIC2 is expressed in the late streak node, which is shown here in this section. And this node expression is transient, and it's gone by this slightly later stage of gastrulation. Strikingly, another family member, ZIC3, is also widely expressed at gastrulation, but ZIC3 is never expressed in the node. So not only is ZIC2 expressed in the right place and right time to cause the identified phenotype, but it seems likely that ZIC2 function is compensated for by other ZIC molecules in all regions but the node. So the other piece of corroborating evidence came from Patrick Tan's lab from a series of experiments done by a postdoc called Anne Camus. So Anne performed some ablation experiments in which he started with an early bud stage embryo. First, She removed the pre-chordal plate cells but left the cranial notochord intact. And when these embryos were cultured for 38 hours, she found their brains developed relatively normally because the pre-chordal plate had regenerated. In contrast, when she left the pre-chordal plate intact and removed the cranial notochord and then cultured these on, she found that pre-chordal plate development failed. So together with our work, which provided the genetic proof, this identified a new inductive process in which the cranial notochord sends a survival signal to the pre-chordal plate. And this has led us to propose this model of pre-chordal plate development. So first of all, the cells pass through the anterior primitive streak and differentiate into pre-chordal plate cells and migrate to the anterior of the embryo. So the anterior primitive streak, under the influence of nodal signals, then induces node formation. And cells that migrate through the node differentiate into anterior notochord cells under the influence of the ZIC2 transcription factor. They then migrate along the midline, coming to lie posterior to the pre-chordal plate cells. Last, the anterior notochord cells send a signal to the pre-chordal plate cells, which is essential for pre-chordal plate maintenance. So, as you can see in this slide, a signaling pathway that's very important for mesoderm formation, midline mesoderm formation and pre-chordal plate formation is a nodal signaling pathway. And what we went on to show, which I won't have time to go into in detail today, is that ZIC2 transcription factor actually acts as a component of the nodal signaling pathway. And one of the target genes of ZIC2 and nodal signaling is FOXA2 expression. And this is important for the last part of the talk that I want to go through relatively quickly. It's our more recent work and unpublished data and some of the quite new experiments. So what we've now been looking at is trying to understand how environmental factors are contributing to HPE disease causation. And we're doing this because so many cases of HPE have no obvious genetic cause. Now, one obvious candidate is alcohol, and that's because we've long known that alcohol exposure early in pregnancy is associated with midline defects and often with HPE-like phenotypes. So this slide shows the experimental protocol that we're currently using to investigate whether alcohol can interact with a small deficit in ZIC2 function to cause holoprosencephaly. So first of all, we use a wild-type female, and she's mated with a male heterozygous for a null allele of ZIC2. And this means that she'll be pregnant with embryos that are either wild-type for ZIC2 or heterozygous null for ZIC2. And then at 7 days of gestation, the female then receives IP injections of ethanol, and we use this particular dose of ethanol, giving two by IP injections 4 hours apart. And then once we've done this, we then dissect the embryos at various time points after the alcohol injection and look for the effects. Now, people always ask us what this might correlate to in humans, and this slide shows the experimental protocol. Shows that mice actually metabolize alcohol quite differently to humans. And the dose required to get the same peak blood alcohol concentration in mice is actually higher than the dose required in humans. And when we do lots of fudging around and calculating, we think that what our model represents an acute moderate dose ethanol model, and that the dose that we're giving is equivalent to a blood alcohol concentration in humans of 0.1%. Now, that's twice the blood alcohol, the drink driving limit, but it actually doesn't take that many drinks within a couple of hours to reach this type of limit in humans. So if we go back to the mouse experiments, when embryos recovered from these females at gastrulation, just some 12 hours after the alcohol injections, what we find, and we look at the node formation using expression of FOXA2 as a marker, we find that embryos that are heterozygous for ZIC2 mutation and exposed to alcohol have aberrant node formation, and they have decreased expression of FOXA2. So in effect, these embryos look very similar to the ZIC2 homozygous embryos. Now, again, this suggests that the nodal pathway is involved, and so we've gone on to look at this using a antibody to phospho-SMAD2, which is a marker of activation of the nodal pathway. And when we do this, we again find that embryos that are heterozygous for ZIC2 and have been alcohol exposed, that these embryos essentially have very little to no nodal signaling, and we find this in all of the treated embryos. And additionally, we know that when embryos recovered from these females at 9.5 days and examined for midline formation using expression of sonic hedgehog, we find that the heterozygous embryos, alcohol treated, again have these phenotypes reminiscent of holoprosencephaly, and indeed, again, they're very similar to the embryos that are homozygous null for ZIC2 in that, and particularly with respect to their brain defects. So we conclude from this that it appears that ZIC2 dominantly enhances alcohol teratogenesis to cause holoprosencephaly background. And in a ZIC2 heterozygous background, alcohol perturbs nodal signaling during anterior notochord production. And we think that ZIC2, mutant alleles of ZIC2 that on their own would not cause holoprosencephaly could dominantly enhance alcohol teratogenesis in humans as well. And with that, I'll finish up and just acknowledge some of the people who've done this work. Most of the work that I've shown you today is being done by Kristen Barrett within my a postdoc within my lab, and Hannah Glanville-Jones, a PhD student in my lab. Thank you.